Contraction of skeletal muscle. What evidence supports the sliding filament mechanism? How does the sliding filament mechanism cause contraction and relaxation? Where does the energy for muscle contraction come from? So, the contraction of skeletal muscle involves two proteins, a thin protein called actin and a thicker protein called myosin. So the actin and myosin filaments slide past one another, and that is what is known as the sliding filament mechanism. So what evidence have we got for the sliding filament mechanism? Well, the myofibrils appear darker in colour where the actin and myosin filaments overlap, and lighter where they don't overlap. So if the sliding filament mechanism is correct, then there should be more overlap of actin and myosin in a contracted muscle than in a relaxed one. So when a muscle contracts, the following changes occur at the sarcomere. So the I band becomes narrower, the Z lines get closer together, which shortens the sarcomere. The H zone becomes narrower. The A band remains the same width because the width of the A band is determined by the length of the myosin filaments. So therefore, it's obvious that it's not the myosin filaments that have become shorter, it's just the fact that the I bands have slid over the A bands. The actin filaments have slid over the myosin filaments. So there's three main proteins that are involved in the sliding filament hypothesis. So myosin is made up of, the thicker myosin is made up of two types of protein. It's got a fibrous protein, which is arranged into filaments, which is known as the tail, and then it's got a globular protein which forms into two bulbous heads. So that's the head. So the myosin has the fibrous tail and the globular head. The actin is a globular protein whose long chains are twisted around one another to form a helical strand. And then you've got this red protein here, which is tropomyosin, which forms long thin threads which are wound around the actin filaments. So in the sliding filament mechanism, the myosin filaments have globular heads that are hinged, so they can kind of tilt back and forward. So it's on a hinge. And each has a binding site for ATP. And on the actin filaments, they have binding sites for the myosin head to bind. And they also have two other proteins, tropomyosin, shown here in pink, and troponin, shown here in green. And they are found between the actin filaments. And these proteins are attached to each other, so the troponin holds the tropomyosin in place. So for myosin and actin filaments to slide past each other, the myosin head needs to bind to the actin-myosin binding sites. So this head needs to bind to the site. But in a resting muscle, the actin-myosin binding site is actually blocked by the tropomyosin molecule. So therefore, the head can't bind and the myofilaments can't slide past each other because their head can't bind to the actin. So, when an action potential arrives from a motor neuron, this stimulates a muscle cell and it depolarizes the sarcolemma. Then the depolarization spreads down the T-tubules to the sarcoplasmic reticulum, which then releases calcium ions into the sarcolemma. And that influx of calcium ions into the sarcolemma triggers the muscle contraction. So the calcium ions then bind to the troponin. So this red bit here is the calcium, the troponin is the green, and that causes the troponin to change shape. And what that does is it pulls the tropomyosin out of the way of the actin myosin binding site on the actin filament. And therefore, the binding site is now free to allow the myosin head to bind. So then a bond forms between the myosin head and the binding site on the actin, and it forms something called an actin-myosin cross bridge. So the calcium ions also activate the enzyme ATPase, which breaks down ATP into ADP and PI, and that provides energy for muscle contraction. And the energy released from the ATP moves the myosin head in like a power stroke to the side, so it tilts the head to the side, which pulls the actin filament along, a bit like a rowing action. So ATP also provides the energy to break the actin-myosin cross bridge, so the myosin head detaches from the actin filament after it's moved, and then it tilts back to its starting position and reattaches to a binding site further along the actin filament. So then a new actin-myosin cross bridge is formed, and the cycle keeps repeating itself. So the head will tilt back, detach, go to the next binding site, etc. So many actin-myosin cross bridges form and break rapidly, pulling the actin filament along, and this overall shortens the sarcomere, causing the muscle to contract. And this cycle will continue for as long as calcium ions are present in the sarcolemma, and as long as they're bound to the troponin. But then, when the muscle stops being stimulated, it goes back to its resting state. So calcium ions leave the binding site on the troponin molecule, and they are moved back in to the sarcoplasmic reticulum by active transport. Obviously, because it's active transport, this requires ATP. So the troponin molecule then returns to its original shape, which then pulls attached tropomyosin back to block the actin-myosin binding site again. So when the muscles aren't contracted because there's no myosin heads to attach to the actin filaments. They can't do it because it's blocked by the tropomyosin, the binding sites. So the actin filaments just slide back to the relaxed position, and this again lengthens the sarcomere back to its resting state.